Hello friends and family, and thank you again for coming to another UC San Diego Create STEM 10 Plus series. My name is Arturo Archibasa, and I'm here to help you out on how to be successful in science. Today we're going to be talking to you about strawberry DNA extraction. How cool is that? Strawberry DNA extraction. Does anybody know what DNA stands for? I didn't, so I just learned. So, what DNA is basically a short acronym for deoxyribonucleic acid. It's something that's going to be pretty long, so we rather use DNA. So the reason why we are picking strawberry today is because strawberry is one of the fruits that has a lot of DNAs. It's actually an octoploid. What that means is that it has 8 sets of chromosomes, like 8 copies of sets of chromosomes, which is a lot. Very a lot. Like for us humans, we are a diploid, so that means we have 2 sets of chromosomes. So that's the reason why we're picking strawberry, but also is because it's more easy to work with, especially in our home lab. Okay, so get excited. We're gonna be doing some pretty cool stuff, and also we're gonna be having Kevin on this video. It's gonna be helping you with the process of the experiment later on the road. I'm here to tell you about the science behind it and what each component that Kevin's using, each part of the material, how is it gonna add to the science behind it. Okay, so stay tuned. Before we get started with this science video, I want to encourage you to go back and watch our video on cells, because it's going to be very important to talk about the components of the cells, because I am not going to go too much into the cell components, so I'll encourage you to guys go into that video and be familiar yourself with the components of a cell, either from plants or animals. But today we're going to be talking about strawberries, so I encourage you to learn yourself more about plants. Well, as you might know, DNA is present in every cell of either plants or animals, and then this allows you to identify the genetic traits, the characteristics of whatever the organism might be. Okay, so what's happening? We're going to be putting some strawberries into a Ziploc bag, and we're going to be pre-smushing them. So it's going to allow us to help break the cells, because what we're going to do, we're going to make a solution that is salt with soap, and then we're going to put it into the smushed strawberries. And what's going to be happening, the soap is going to be basically dissolving the membrane of the cell. And then what the salt's going to be doing is going to be breaking those protein chains for the nucleic acid to be able to extract the DNA. And then one thing that you want to do before you do this experiment is to have some ethanol, some rubbing alcohol, be chilled. You want to have it cold, because the more colder it is, the DNA will precipitate, because the DNA is not soluble in like rubbing alcohol, ethanol, or isopropyl alcohol. And it's not, so what happens, it precipitates, and then this allows it to rise up and come up and be able to see it. Okay, so stay tuned. We're going to get started, and here we go. Take it away, Kevin.
Hi everybody, my name is Kevin. Today we'll show you how to extract DNA from a strawberry. Here are the materials needed for the experiment. Strawberries, liquid dish soap, coffee filters, 70% rubbing alcohol. Make sure to get it cold before the experiment. Salt. Instead of sticks, we're using spoons. Clear plastic cups, Ziploc bags, disposable gloves, or an apron or a lab coat, goggles, jar, and a large mixing spoon. First, we'll create a dish soap salt solution. Add 1/3 cup of warm water. Add 1 tablespoon of dish soap. add 1/2 tablespoon of salt into the pitcher. Second, we will be putting 2 strawberries without the stems into the Ziploc bag. Begin to smush the strawberries without making any holes into the bag. Next, we will grab our salt solution, add a small amount like 3 tablespoons, and continue to smush the strawberries in the Ziploc bag without making any holes. I'm going to transfer this liquid to this beaker to make it easier for the next step. Time to drain. Grab a coffee filter and carefully pour your solution of the Ziploc bag into the filter and allow it to filter into the clear cup without coffee filter ripping or big chunks passing through. Make sure that the solution in your cup is mostly liquid without no really big chunks. You don't want any pulp. Lastly, very carefully pour the chilled 70% alcohol into your cup, but do this slowly by tilting your cup. You are not trying to mix here. Now I'm going to show you the DNA.
Well, that's all we have for today, folks. Thanks again for checking us out. You know, here at UC San Diego Create, we love to support everybody in education, especially in STEM and sciences. Another thing that we want to go ahead and give you a message is to stay safe out there, wear your mask, make sure you wash your hands, and that's pretty much all. Check us out for next week's lesson video. Thank you very much. Have a good one.